Hey there, this is Dr. Fu. This module, we're going to talk about neuromodulator drugs, and we're going to use cricket to test the effect from different drugs. And this is the objectives. Particularly, we're going to learn some effects from neuromodulator drugs. And then we're going to use our Power Lab, which is a very fancy machine, to record electrical signals from neurons so we can learn the properties of these electrical signals. Okay, before we get started, I have some questions to ask you. Here's one. We all know resting membrane potential is about negative 17 millivolts, or some textbook will say negative 75 or negative 65. But all of them are negative, is that right? What does this negative sign here mean? Have you ever think about that? Before we understand that, we have to go back some physics. First one is the battery. So this is the anatomy of the battery, and this inserting part is a positive end. And inside this positive end, there are a lot of positively charged particles. And in this surrounding area, that is negative end. And in this negative end, we have a lot of negative charged particles. And the positive and negative particles, they never meet, because the membrane wrapping around the insert is going to prevent that. However, if you connect a line here, you are going to move some negatively charged particles away from the negative end and go to the positive end. Here is the situation. You have battery and then you connect a line, then the negative charged particles, mostly that's the electrons, will move this way back to your positive end. And if you put a voltmeter connected here, you are going to measure the voltage of this battery. Okay, here's a question. How does the multimeter works? Okay, so a voltmeter is a large resistor, and we already know the resistance of it. Why it has to be a very large resistor? Because most of the case, when you're measuring voltage, you are going to connect your voltmeter into a battery, and then if the resistance is not large enough, you are going to have a lot of current go through, then that's going to damage your voltmeter. So all the voltmeter will have super large resistance. And then because we have this Ohm's law, we know the resistance of our voltmeter, then we can measure the current that goes through the voltmeter, then we can calculate the voltage that we're going to measuring. If you connect your battery and your voltmeter this way, the electron is going to enter the voltmeter from this black end, I call this reference end, and then exit through this red end, I call this measure end, and then you are going to get a positive voltage. However, if you connect your voltmeter the other way, the electron is going to enter your voltmeter from this measure end and then exit from your reference end, and you get a negative voltage. So whenever you see a positive sign or a negative sign from this voltmeter, you will know if that's a positive sign, that just means the measuring end is connected to a spot that has more positively charged particles. For example, that is positive, and then the measure end, and that end is connected to a place that have a lot of positive charge particles. That's why electrons are going to flow towards this direction. And if the voltage is negative, then you know the reference end is connected to a spot that has more positive charge particles. So this situation, this is negative, so you know the reference end is connected to a place there are a lot of positively charged particles, so the reference end is pulling a lot of electrons towards it. Okay, with that being said, let's come back to our cell. I have a reminder. If the sign that you get is negative, which means the black end, which is the reference end, is connected to a place there are a lot of positively charged particles, and this, in this situation, is negative, so you know there are more positive particles in here, less positive particles in here. When people measure the voltage of a cell, they always connect the black end, which is the reference end, outside the cell, and then the red end, which is the measure end, in the cell. Alright, I got something more to confuse you. Okay, so early on we talked about the electron will flow from the negative end, go through all the way to the positive end, right? And that is how electron, the negatively charged particles, flow. And the definition for current is the opposite direction of your electron flows. So actually the electricity, the current, flows just the opposite, like this way. And that is called conventional current. Okay, let's apply our electricity knowledge. Okay, so here is called multimeter. So a multimeter can measure voltage and resistance or other things. So we'll use multimeter to measure the electricity from a wall outlet. And I'm just going to explain you how to do that. Don't try to do that on yourself. If you shock yourself, it's not my fault. Okay? All right. So all the wall socket that will have 3 holes. So one of them is neutral, one is ground, and one is hot. If you plug in your measuring end, which is the red end, to this relatively small hole that is called hot hole, and then the black end to this neutral hole, you get 120-ish volts. That means this red thing is connected to a place there are a lot of positively charged particles, right? So behind this hole, it's going to connect it to a place you can find a lot of positively charged particles. And the same thing, if you connect your multimeter to the ground hole, you get very similar number. That's because this ground hole and neutral hole, they have similar functions. I'm going to show you how that's the case. Okay, so here's how do you get your electricity. This is a power plant, and then you get your positively charged articles located in here. If you connect the line there through, let's say if this is your multimeter, and then you connect it to your neutral hole, which is this hole, and then your positively charged place is going to pull a lot of negatively charged article, which is electron, from the neutral end all the way into your multimeter, and then your multimeter will read number and then send back these electrons to this positively charged place. And the same thing for your ground. If you connect your multimeter, the red end to your hot, the other to the ground, you are going to have a lot of electron put out from the ground, which is the earth, go through your multimeter and go all the way to your positive end, which is through this hot hole, right? And that way you will read the numbers, the voltage between your power plant and then the ground. Actually, the neutral and the ground, they kind of like back up each other. Whichever provides a lower voltage is going to close up your circuit. Well, actually, when you plug in your appliances, for example, a washing machine, you have to plug in all these 3 holes. The electricity either flow from hot to your washing machine, go back to neutral, or from the hot washing machine to ground. Okay, let's come back to this neurotransmitter, neuromodulator topic. Let's talk about some terminologies. First one is neurotransmitter. That is a messenger, and most of the time that is a chemical that released from a neuron and which will diffuse across synaptic cleft to generate an immediate short-term effect. And you remember what is synaptic cleft is, right? That's where 2 neurons meet. It's a very slit, very small, narrow place. And the effect is only on the post-synaptic neuron. And then what is a neuromodulator? A neuromodulator is a messenger, which is also a chemical that will be released from a neuron and not necessarily at the synaptic site, which means it can release anywhere. And these neurotransmitters will affect a group of neurons as long as these neurons have appropriate receptors. Okay, and then the question is, what is a receptor? A receptor is a molecule that receives signals. Okay, so most of the time that will be a protein, and most of the time they are on the surface of the cell, so they can receive signals from the environment. Can be your internal environment. For example, your blood vessel cells have receptors to regulate the tension of the blood vessel, or can be from the external environment. For example, the eye, the retinal neurons have photoreceptors, can receive signals from the light. So anything that will receive signals from outside cell and report that into the cell, we call them receptors. Okay, there are 2 types of receptors in the neurons. One happens really fast, the other is kind of slow. So when we're talking about fast, it happens in milliseconds. So you will have 1,000 milliseconds in 1 second, is that right? So these things happens really fast. And the other type happens a little bit slower, and we're not talking about it happens in 10 years. These things will be activated in second or even a minute. So they're slow, but they're not that slow. The fast type of receptors are usually the ionotropic receptors, and they are transmembrane proteins. Trans means across, so these receptors, they are embedded on the surface of the membrane. So most of them are just ion channels. They can open and close like iron, go in and out. These things happen really fast. And the other type is metabotropic, and they are usually the transmembrane proteins that will initiate signaling cascade within the cell. So this is a metabotropic receptor that is a protein. It is embedded in the membrane, and then once this protein receptor get activated, it's going to signal the downstream proteins. Most of the time, the downstream proteins are G proteins. So this type of receptors are also called G protein coupled receptors. And once these G proteins are activated, they are going to initiate a signaling cascade downstream of these G proteins. So there's going to be a chain reaction that happens after this. And because this type of receptor will require a reaction of several proteins together, so this type of receptor will take time. Okay, next, let's talk about the type of neuromodulators. There are different ways to categorize the neuromodulators. The first one is based on the effect of these neuromodulators compared with their neurotransmitters. So agonist and antagonist. Agonist is the neuromodulator that has a similar effect or will facilitate the effect of neurotransmitter. And antagonist is the neuromodulator will have just the opposite effect of the neurotransmitter. So these are the enemies of these neurotransmitters, and these are the friends, the helpers of these neurotransmitters. Another way to categorize a neuromodulator is excitatory and inhibitory. So excitatory neuromodulator is going to excite a neuron. Inhibitory neuromodulator is going to inhibit a neuron. So a agonist doesn't necessarily to be a excitatory modulator. Am I right? If the neurotransmitter is inhibitory, and then the neuromodulator that is an agonist of this neurotransmitter is going to be an inhibitor too. Is that right? Okay. And the third way to categorize these neuromodulators is competitive and non-competitive. So what does that mean? So competitive neuromodulator means these neuromodulator will compete for the same binding site as their neurotransmitter. So here is the example. This is the binding site of the neurotransmitter. And then if you have a neuromodulator, which is a drug, they also bind the same site here. So there's only one place to sit. They compete for this site. And in the non-competitive, they bind to different sites. So this is a non-competitive. This is a neurotransmitter binding site, and this is the drug, the neuromodulator binding site. Okay, and both the competitive and non-competitive neurotransmitter, they can be either agonist or can be antagonist. It's not necessarily always competitive is agonist or competitive is always antagonist. Okay, what other information you can get out if I'm telling you a neuromodulator is competitive? Here's the thing. If I tell you there's a lot of neurotransmitter in your system already, which means all these binding sites for these neurotransmitters are all occupied, okay? And if you put more competitive neuromodulator to the system, you are not going to see any further effect because you don't have any binding site for the receptors, right? So the competitive receptors, you have to consider if these neurotransmitters already saturate all your binding site of these receptors. And for the non-competitive, you don't have to worry about that because even these binding sites by these neurotransmitters are saturated, which means they all occupied, you still have more binding site for these neuromodulators to bind. Because remember, all these neurotransmitters, they can only bind to this binding site. They don't bind to these binding sites. So you are still be able to have further effect, even these binding sites are saturated. Okay, now let's talk about how we're going to record neuron activities. There are two types of neuron activity recording. First one is single neuron potential recording, which means you only record action potential from one neuron. And there's a local field potential recording, which means you record activity of a group of neurons. For this single neuron activity, you can only see the frequency change because for one neuron, the amplitude of the action potential never change. They stays pretty much the same all the way, right? So you can see the action potential sometimes is a little bit higher, sometimes lower, but majorly maintain the same level. And then let's review our electricity knowledge. So this is the voltmeter, and then this is the reference end, this is the measuring end. And then if you get a negative voltage when the cell is resting, so resting membrane potential is negative, that means your reference end is in this place that have a lot of positive new particles, and the measuring end have less positive particles. All right, let's back to this single neuron potential recording. This is a healthy subject. You can see their neuron fires at a certain frequency, and this is how the neurons from Parkinson's disease patient fires. They fires relatively with lower frequency. And for local field potential recordings, because you are recording a group of neurons, so the number that you recorded for the frequency and the amplitude, they both may change. However, the frequency measure is artificial. I'm going to show you why just in a second. Okay, so here's an example. How do you read amplitude of the frequencies? So this is the time versus the voltage of the field potential recording, and you see in this box and in this box the amplitude is how much the voltage will go up and down. See the area that green covers compared to this time, the second part is the amplitude is higher than the first recording. Is that right? Okay, let's talk about the frequencies. If you zoom in from this graph, that is around 55 seconds, and you see that's 55 seconds until 55.1 seconds. So this 1.1 second, you have a lot of peaks, and if you measure how many peaks you have divided by the time, which is 0.1 second, you get the frequency of these firings. However, you can say, okay, I'm going to roughly combine these peaks together. I get peak here, get peak here, then the frequency will drop. So the frequency measurement is actually based on your setup a threshold for your peak. For example, the peaks here, you can say, okay, I consider them as just a noise. They are component for this big peak. So when they are forming this big peak, they have a little bit noise going on. So whether you consider these peaks as a peak, it's artificial. So that's why I say this local field potential recording, the frequency measurement is artificial. All right, next let's talk about spontaneous firing and stimulus evoked firing. So spontaneous firing. These firings are from neurons that are responsible to maintain the basic body functions, and they are always on. For example, we also have these spontaneous firing neurons. They belong to autonomic system, like your heart rate, your breathing, all these vital functions, your blood pressure. These are maintained by these autonomic neurons. And then in the cricket, these neurons, we call them spontaneous firing neurons. And then the stimulus evoked firing, they are the neurons that respond to the environment. So they only fire when they get stimulated. For our case, our somatic neuron, the sensory neuron, the motor neurons who responds to the environment, you feel things, you do things. These neurons are stimulus evoked fire neurons. So the cricket, they have these kind of neurons too, and they are only fired when they get stimulated. So the spontaneous firing and stimulus evoked firing, they are actually from different neurons. Okay, now let's take a look at the examples of the recording from a cricket. This is the same image that I showed you earlier. Same thing, this is the timeline, and this is the voltage. So you can tell the amplitude for the height of each firing. And then the red rectangles circles out the spontaneous firing. Okay, so during this time, I didn't do anything to the cricket. They just lay there and they have some neurons that's firing. So that's spontaneous firing. And then in this blue boxes, I did something to the cricket, which is not very pleasant, I guess, to them. I poked their cerci. I won't tell you what is cerci. So you see there is some firing here, and these bigger spikes, these are from stimulus evoked neurons firing. And then same here, the lines here are crazy. It's because I inject the solution to the cricket. So there's a lot of stimulus happening. That's why they get a lot of neuron firing. You see the amplitude change a lot because there are more neurons get fired during this short period of time. And then after I take my needles out, stop my injection, they come down, and then they back to this spontaneous firing. Okay, next let's talk about some examples of neuromodulators. Okay, the first one is nicotine. So nicotine is a competitive agonist to acetylcholine. So that means nicotine is going to bind acetylcholine receptor at the same binding site as acetylcholine. That's what is a competitive mean, right? And agonist means nicotine will have the similar effect as acetylcholine, or nicotine is going to facilitate acetylcholine. And what is acetylcholine do for insect? Acetylcholine is a main neurotransmitter in insect that will lead to neural excitation because it will increase Na influx. If you have Na go into your cell, then the voltage inside of the cell will increase because Na is positively charged, right? So that will make your neuron easier to reach the threshold to have action potential. Okay, so an insect, if they exposed to high concentration of nicotine, they are going to have seizure and die. Okay, so here's a question. Can any insect live on tobacco plant? Why do I ask that? Because to get tobacco plant have a lot of nicotine, and then the insect live on the tobacco plant. If they eat the tobacco plant, they will die. Is that right? Then you probably will think, okay, then no insect can live there. Well, actually, the answer is yes. Why is that? Because some insects don't have this receptor. So you see how important to have a receptor to respond to certain drugs, right? And this is how nicotine works. So this is a spontaneous firing, and then after you inject the nicotine into a cricket, so they take out the recording when they're doing the injection. So this is when the cricket calm down. So you can see the spontaneous firing get increased a lot. So that's why nicotine in insect is a excitatory neuromodulator, right? And I want to ask you for this recording, is this field potential recording or single neuron recording? The answer is this is a field, right? Because you see the amplitude change. So here is how acetylcholine and nicotine work in the insect. So this is the neuron of the insect. On the neuron, you have this acetylcholine receptor, and that receptor will bind either to acetylcholine or nicotine. And then after they bind, they are going to let Na get into the cell, and this neuron is going to be fired. Okay, next one is GABA. I said the GABA bind to this receptor. That's too long, I'm not going to say that. And then in human, the GABA binding will lead to neuron inhibition because you are going to increase chloride into the cell. Chloride is negatively charged, so you are lowering the voltage inside the cell, and then you are going to have harder time to reach threshold to have action potential. That's why these neurons get inhibited. So GABA is a inhibitory for human. And GABA also have a lot of agonists, and these agonists are not competitive agonists. For example, general anesthetics. So these anesthetic drugs put you asleep. They are also the GABA agonist. So they inhibit your neurons, so you're gonna fall asleep. And there is the other binding site. They all can inhibit your neurons. Actually, alcohol is also a non-competitive agonist to GABA. See here, the ethanol, that means alcohol. That's a scientific name for alcohol. So they also bind to this binding site and have a similar effect as GABA. So here I said it's a non-competitive to GABA. They can lead to neuron inhibition in human. I said they can. It's not necessary always. Depends on the amount of that you drink. For example, if you had experience with alcohol, I'm sure you did, huh? Sometimes if you get overdose on alcohol, some people get over talkative. You see those people, they get excited, and some people they get depressed. They will just go back to sleep. So that really depends on different people. However, some certain neurons will be inhibited after you have alcohol consumption. For example, the judgmental part of the brain most of you guys after drinking alcohol, that part get inhibited. That's why when after you have alcohol, some people doesn't look that nasty anymore. Like you have like people that you hate and then you go drink together with them and those people looks so lovely after alcohol, right? Okay, so that's because the judgmental part of the brain gets suppressed, and also your fine motor movement neuron gets suppressed too. That's why you're not allowed to drive cars after you have alcohol, because some of the motor neurons get inhibited, so you won't be able to respond to emergencies that fast anymore. Okay, next one is glutamate. I said glutamate is a powerful excitatory neurotransmitter, and it's released by neurons in your brain, and then they need to be cleaned up by astrocyte. If don't, these extra glutamate circulating in our brain is going to cause neuron death. So we call this kind of that glutamate excitotoxicity, because these neurons get too excited from this glutamate that they exhaust themselves and then they die. Okay, if glutamate is that toxic, is monosodium glutamate toxic? Basically, they're the same thing. The monosodium is just the glutamate plus a sodium. So once these things get into water, it's going to split into a sodium and a glutamate. We also call them MSG. Chinese people use MSG pretty much every single meal they cook. I mean, I use it. But the question is, we eat so many MSG, but we didn't die. Why is that? Because Chinese people have superpower? No, because we have brain-blood barrier. The MSG that we eat will go into our gut and then get absorbed and then will enter the blood circulation. Will probably go to your tissues but not to your brain because of the brain-blood barrier. Okay, and then the MSG is that inhibitory or excitatory in cricket. We're gonna do experiment to test it. Okay, let's talk about how do we record neural activities in our lab. First of all, we're going to use cricket, and then we're going to put the cricket with their ventral side up, which means their belly up. And because their nerve cord is very close on their ventral side, so it's easier to pin to their nerve cord if you put them belly up. If not, then it's hard to get signal. So you're going to put 3 pin electrodes in there. We call them lead wires. And these lead wires are going to be connected to a machine called PowerLab. And that basically functions as a multimeter, which will measure the voltage. So there's this big nerve here, and then all these 3 pin electrode, the lead wires are going to connect it to this nerve cord, which is this guy here. And then 2 of these electrode functions as a reference. You remember in the wall socket, there's 2 of them actually functions as a negative end. One is neutral, the other is ground, right? So whichever is lower, we're going to use that. Same thing here. So if the neurons in this region get activated, so which means inside of this neuron is going to have higher voltage, more positively charged particles, right? And then the electrons either from here or from here get pulled into this site. And this electrode basically functions as our measurement end of the multimeter. And because this measuring end is connected to an area that has more positively charged particles, so our multimeter will have a positive reading. And you can inject different drug and observe different drug will cause different changes of voltage. Okay, so what about this situation? If the neurons in this end, they become excited, the machine will read this one as negative because the machine is set up by default. As long as one of the reference pin has more positive charge particles than the measuring end, is going to read a negative. This situation is going to become positively charged because the machine is set up by default. As long as the measuring end has a more positively charged particles than one of the reference pin, it's going to be set up at positive. So you have to understand, from these neurons get activated till this neuron get activated, or both of them are get activated, these things happens really fast, like in millisecond. So they are not going to maintain this status for a long time, for even 1 second. So they will quickly go from this neuron activated to this neuron activated, or both of the, or even here get activated. So that happens really fast. So the way we set up our Power Lab to read voltage is a little bit complicated. You can program that to read differently. However, this way we're more error-proofing. If one of the electrodes didn't get pinned right into the center of the nerve, they're a little bit off, we can still have readings. And about how to set up the Power Lab readings, that can be another whole semester's lab, and we don't have to dive that deep for this course. Okay, last thing is something about the anatomy of a cricket. So the cricket will have two cerci, or for singular form, each of them is a sulcus. On their cerci, they have little hairs, and the bottom of these little hairs, they have their mechanoreceptors. Do you have mechanoreceptors? Yes, you do. Where you find them? At the bottom of your hairs. So even if you're not looking at your hairs, somebody touch your hair, you will be able to feel that. Is that right? That because at the bottom of your hair, you have these mechanoreceptors. Will sense where the hair move. So does the cricket. If you use a wood stick to touch their cerci, this here, they are going to feel there is some force coming this way. And for some female cricket that have the third tail, which is the ovipositor, that is how they lay their eggs. So this is not their sensory organ. So when you're stimulating their cerci, you want to go a little bit fast. You don't want to approach them and stay there. They're going to get used to that and not respond to you. And the best spot to stimulate that is the inner side part. So here is the most sensitive spot. Alright, this is the lecture part. Okay, I'll see you in the experimental video. Bye bye.